Hey everyone, my name is Hannah Zang, and today I'm going to be talking about conducting molecular oncology research. I've worked on this project with Dr. Alexander Beezer for the past 3 years, and I hope that this presentation will help explain what I've been working on. So let's get started. So here we have a picture of both beer and bread. I know that many people love both of these things, but I want you to think about what they have in common. And if you've ever walked by Dr. Beezer's lab in Derby, where he makes beer, you may think that it smells a lot like bread, and that's because bread and beer both use yeast. So yeast is what I actually use in my research, and yeast is also known as Saccharomyces cerevisiae, but I'm going to stick with calling it yeast. So here we have a microscopic picture of yeast, and yeast have a lot of friends, so they like to stick together and form colonies, and the size of these colonies can provide insight into the growth rate, but more on that later. So when I'm talking about using yeast in science, I'm using yeast as a model organism, and here's a picture of some model organisms. So model organisms are living organisms that we use for scientific research. It turns out that research in humans can be pretty complicated, so that's why we sometimes prefer to use model organisms to humans. So next what we're going to talk about is the central dogma, and we're going to try and break down this foundational concept of biology. So maybe you know that what makes us unique is our DNA, and the DNA will undergo a process called transcription to become mRNA, and mRNA will undergo a process called translation to become proteins. So if you're confused, try and think of transcription and translation in a different way. For example, your transcript is a record of all of your grades, so it just keeps a copy of the grades that you've made. And translation, on the other hand, is becoming an entirely new language, like mRNA when it becomes proteins. So you may be thinking of proteins, like proteins in our diet, such as nuts and eggs, but I'm actually talking about biological proteins. So one biological protein is called hemoglobin, and it carries oxygen so that we can breathe. It turns out we have a lot of proteins, and they all help us in different ways. So what exactly makes biological proteins? Ribosomes. And ribosomes, you can kind of think of them as like complex little factories. So here's a diagram of the different ways to represent ribosomes. And in the next few slides, I'm actually going to start talking about my research, which focuses on a small part of how we make ribosomes. So here's the entire process of how we make ribosomes. And I don't want you to think this is a simple process. It's actually very complicated and probably a bit overwhelming. But don't worry, we're not going to talk about it in this much detail. Instead, we're going to focus on the part that I worked on in my research. So the specific protein that I work on in my research is called YVH1, which is a type of trans-acting factor. And you can think of trans-acting factors as proteins that like to regulate everything to make sure that the process occurs smoothly. So maybe you can think of them as road signs because they give us direction. Now here I'm going to represent YVH1 as an orange oval, and the blob shape is the ribosome. So YVH1 works on displacing another protein called MRT4. So with YVH1, you can see that the green oval called P0 can attach to the ribosome. So without YVH1, we find that MRT4 remains bound to the ribosome, and since it has what we call a higher binding affinity, the P0 cannot bind to the ribosome. So here we have an X to show that it is not binding, and I'll try to explain what I mean by binding affinity in the next slide. So, just to recap, with YVH1, the P0 can attach to the ribosome, and without YVH1, P0 cannot attach. So to explain binding affinity, try to think of MRT4 kind of like Gorilla Glue, and P0 as Elmer's glue. So MRT4 will stick to the ribosome way more than P0. Now, proteins are all composed of amino acids, which are like building blocks, kind of like Legos, and they're all different, so the pattern of these building blocks can create different protein shapes. So YVH1 has 364 amino acids, and we can split it up into two parts, with the second part being what helps YVH1 displace MRT4. Okay, so now what we can do is we can use a process called polymerase chain reaction, or PCR. And we can make sure that this process is error-prone, which is like the opposite of Grammarly. We're like introducing mistakes into the second part of YVH1. And it turns out that there are different types of mistakes, which we call mutations, that we can introduce. So here are 3 types of mutations. The first is that we can create an entirely new amino acid, which can change the shape of the protein. The second type is where we can create a red light, or a stop codon, and this stops the rest of the protein from being made. And lastly, we can have a silent mutation, which there isn't an effect at all and the protein keeps its shape. Now I mentioned earlier that yeast colony size can indicate growth. So when there is no YVH1, the yeast actually grow a lot slower and the colonies are a lot smaller, as you can see up here. But we also want to see what type of mutation was introduced, whether it was a changed amino acid or a red light. So since red lights stop the rest of the protein from being made, they're a lot smaller than the normal protein. And we try to measure these proteins by what we call Western blot. So on the right is a Western blot that I ran, and you can see a ladder to measure the proteins in the orange. So this is how we determine the type of mutation that is introduced. Now, why is all of this important? Well, YVH1 is actually found in humans, and we call this human version DUS12. When DUS12 is overacted, this causes a type of cancer called liposarcoma, shown here. So hopefully this research can help us figure out how this cancer exactly works, and we can potentially work on drug therapies in the future. So I hope that this presentation helped you understand a little bit about my research project. I would also like to take the time to thank my research mentor, Dr. Beezer, as well as Dr. Buby and Dr. Buell for giving me the opportunity to practice this Pika-Kucha format. I'd also want to thank our collaborators and organizations that funded this research, making it possible.